Welcome to The Explainer. Today, we're diving into a real-life scientific detective story. The mystery we're trying to solve? Figuring out the genetic culprits behind one of the most common birth defects out there. And believe me, the stakes are incredibly high. Just look at this number. That's how many people died from congenital heart disease, globally, in just 1 year, 2017. So yeah, this is a serious mystery, and you better believe scientists are on the case. Okay, so before we can solve any case, we need to know what we're actually looking for, right? We're talking about congenital heart defects, or CHDs for short. Basically, these happen when the heart just doesn't develop properly in the womb. But the million-dollar question is, why does this happen?
All right, let's get into the thick of it, and I'll tell you right now, this is a seriously complex case. We're not talking about some simple open and shut situation with one obvious bad guy.
Yeah, this investigation is tricky because there's almost never a single culprit. The term researchers use is multifactorial. It's like a really complicated dance between our genes on one side and our environment on the other. And what that means for our detective story is that there are a ton of potential leads to chase down. So, where do our scientific detectives start? Well, when a case is this tangled, you always start with your most promising leads. Let's see where the clues pointed first.
It turns out, for a long time, a major area of interest has been something called the folate pathway. Now, you've probably heard of folate, right? We know it's super important for preventing birth defects. So the thinking went something like this. What if tiny genetic variations, little typos in the enzymes that process folate, are what's messing up a baby's heart development? That idea made the genes in this pathway a total research hotspot.
Okay, so now our detectives have a solid trail to follow. They started to zero in on two very specific genetic suspects. They knew where to find them, and they had a pretty good idea of a motive. It was time to pull their files. And here they are. This is great. It's like we've got their genetic mugshots right here. So on the left, you've got MTHFD1. Its MO, a simple letter swap. A G becomes an A. But that simple change can slow down its enzyme. Now look to the right. Our second suspect, CBS, is a bit more dramatic. Its MO is to stick in 68 extra letters of code. This basically throws up a premature stop sign, telling the cell to stop reading the instructions way too early. You can see how both of these guys have a really plausible motive for causing trouble during development. All right, let's get to the actual investigation. This all went down in the Jammu region of North India, where scientists set up what's called a case control study. The whole point was to see if our two main suspects were hanging around the scene of the crime more often than you'd expect. The plan was actually pretty straightforward, but really clever. You take two groups. Group 1, 80 kids who have CHDs, those are your cases. Group 2, 80 healthy kids, those are your controls. Then you compare their DNA. The whole investigation hinges on one question. Do our genetic suspects show up more often in the first group? And here's the game plan, the step-by-step process they followed. First up, you got to get your participants, right? The 80 cases and 80 controls. Step 2, you take blood samples and pull out the DNA. Step 3 is the high-tech part, genotyping. They used lab techniques to specifically hunt for our two suspects, those MTHFD1 and CBS variations. And finally, step 4, you run the numbers and see what you found. You statistically compare how often the suspects appear in each group. So, all the evidence was collected. The data was in, the numbers were crunched. It was time for the final verdict. What did the investigation actually uncover? I mean, this is it, right? This is the one question the entire study was built to answer. Were these two genetic suspects actually responsible for the heart defects? Did they find the smoking gun? Okay, let's look at the evidence against suspect number 1, CBS. So the risk version of this gene, it showed up in 6% of the kids with heart defects. And in the healthy kids, only 4%. Now you might think, hey, that's a difference. But in the world of statistics, that difference was just too small. It wasn't statistically significant, which is a fancy way of saying it could have easily happened by pure chance. You know, like if you flip a coin 10 times and get 6 heads instead of 5. It doesn't really mean anything. It definitely wasn't the strong link they needed. And then there was MTHFD1. The case against this guy? Well, it pretty much fell apart. In fact, if you look at the table here, check out the bottom row for the GG version, that's the suspect variant. You'll see something kind of weird. It was actually less common in the kids with heart defects, only 12.5%, than it was in the healthy kids, where it was 20%. So not only was there no link, the evidence almost pointed the other way. The trail went completely cold. So, what's the bottom line? Let's just go straight to the source, to the researchers' own conclusion. The verdict was crystal clear. For this group of people, in this specific investigation, both of our prime suspects were not guilty. Okay, so at this point, it's really easy to think, well, that was a waste of time, right? They didn't find anything, they proved their suspects were innocent. But here's the thing about science. That could not be further from the truth. And this, right here, is maybe the most important takeaway from this whole story. Getting a not guilty verdict, what scientists call a null result, is not a failure. It is a finding, and it's an incredibly valuable one. Just think about it like a real detective case. By proving these two suspects are innocent, this study basically puts out an all-points bulletin to the entire scientific community saying, hey everyone, for this population, you can stop looking here. This lead is a dead end. That saves so much time, so much money, and it lets all the other detectives focus their energy on leads that might actually pan out. It's a crucial piece of the puzzle that actually makes the whole investigation stronger. And really, that's the beautiful thing about science, isn't it? It's this ongoing story. Every answer you get, even if that answer is no, just helps you ask the next smarter question. So this case, it's far from closed. It just has 2 fewer suspects on the list, which means we're all one step closer to finally solving the real mystery.